Thank you for joining me on this adventure. Your presence means the world to me. Welcome, friends, to our video dedicated to miRNA sequencing. Let's dive right in and uncover its wonders. miRNA sequencing, miRNA-seq, a type of RNA-seq, is the use of next-generation sequencing or massively parallel high-throughput DNA sequencing to sequence miRNAs, also called miRNAs. miRNA-seq differs from other forms of RNA-seq in that input material is often enriched for small RNAs. miRNA-seq allows researchers to examine tissue-specific expression patterns, disease associations, and isoforms of miRNAs, and to discover previously uncharacterized miRNAs. Evidence that dysregulated miRNAs play a role in diseases such as cancer has positioned miRNA-seq to potentially become an important tool in the future for diagnostics and prognostics as costs continue to decrease. Like other miRNA profiling technologies, miRNA-seq has both advantages: sequence independence, coverage, and disadvantages: high cost, infrastructure requirements, run length, and potential artifacts. In the upcoming section, we'll be dissecting introduction and exploring its intricate connections to our topic. miRNAs. miRNAs are a family of small ribonucleic acids, 21-25 nucleotides in length, that modulate protein expression through transcript degradation, inhibition of translation, or sequestering transcripts. The first miRNA to be discovered, Lin4, was found in a genetic mutagenesis screen to identify molecular elements controlling post-embryonic development of the nematode Caenorhabditis elegans. The Lin4 gene encoded a 22-nucleotide RNA with conserved complementary binding sites in the 3' untranslated region of the Lin14 monotranscript and downregulate Lin14 protein expression. miRNAs are now thought to be involved in the regulation of many developmental and biological processes, including hematopoiesis miR181 in mus musculus, lipid metabolism miR14 in Drosophila melanogaster, and neuronal development C6 in Caenorhabditis elegans. These discoveries necessitated development of techniques able to identify and characterize miRNAs, such as miRNA-seq. In the upcoming section, we'll be dissecting history and exploring its implications in greater detail. miRNA sequencing. miRNA-seq was developed to take advantage of next-generation sequencing or massively parallel high-throughput sequencing technologies in order to find novel miRNAs and their expression profiles in a given sample. miRNA sequencing in and of itself is not a new idea. Initial methods of sequencing utilized Sanger sequencing methods. Sequencing preparation involved creating libraries by cloning of DNA reverse transcribed from endogenous small RNAs of size selected by column and gel electrophoresis. However, this method is exhaustive in terms of time and resources, as each clone has to be individually amplified and prepared for sequencing. This method also inadvertently favors miRNAs that are highly expressed. Next-generation sequencing eliminates the need for sequence-specific hybridization probes required in DNA microarray analysis, as well as laborious cloning methods required in the Sanger sequencing method. Additionally, next-generation sequencing platforms in the miRNA-seq method facilitate the sequencing of large pools of small RNAs in a single sequencing run. miRNA-seq can be performed using a variety of sequencing platforms. The first analysis of small RNAs using miRNA-seq methods examined approximately 1.4 million small RNAs from the model plant Arabidopsis thaliana using Lynx Therapeutics' massively parallel signature sequencing (MPSS) sequencing platform. This study demonstrated the potential of novel, high-throughput sequencing technologies for the study of small RNAs, and it showed that genomes generate large numbers of small RNAs with plants as particularly rich sources of small RNAs. Later studies used other sequencing technologies, such as a study in Celsius elegans, which identified 18 novel miRNA genes as well as a new class of nematode small RNAs termed uRNAs. Another study comparing small RNA profiles of human cervical tumors and normal tissue utilized the Illumina company genome analyzer to identify 64 novel human miRNA genes as well as 67 differentially expressed miRNAs. Applied Biosystems' solid sequencing platform has also been used to examine the prognostic value of miRNAs in detecting human breast cancer. As we enter this new phase, let's uncover the impact of small RNA preparation on our broader topic. Sequence library construction can be performed using a variety of different kits depending on the high-throughput sequencing platform being employed. However, there are several common steps for small RNA sequencing preparation. Total RNA isolation. In a given sample, all the RNA is extracted and isolated using an isophycyanic phenylchlorophenol method or a commercial product such as trisyl enriched reagent. A starting quantity of 5100 ig total RNA, 1 g of tissue typically yields 1 g of total RNA, is usually required for gel purification and size selection. Quality control of the RNA is also measured, for example running an RNA chip on Caliper Labchip's Caliper Life Sciences. Size fractionation of small RNAs by gel electrophoresis. Isolated RNA is run on a denaturing polyacrylamide gel. An imaging method such as radioactive 5-labeled oligonucleotides along with a size ladder is used to identify a section of the gel containing RNA of the appropriate size, reducing the amount of material ultimately sequenced. This step does not have to be necessarily carried out before the ligation and reverse transcription steps outlined below. Ligation. The ligation step adds DNA adapters to both ends of the small RNAs, which act as primer binding sites during reverse transcription and PCR amplification. An adenylate single-strand DNA is followed by a doctor is ligate to the small RNAs using a ligating enzyme such as RNA ligase. The adapters are also designed to capture small RNAs with a 5-phosphate group, characteristic miCroRNAs, rather than RNA degradation products with a 5-hydroxyl group. Reverse transcription and PCR amplification. This step converts the small adapter ligate RNAs into clones used in the sequencing reaction. There are many commercial kits available that will carry out this step using some form of reverse transcriptase. PCR is then carried out to amplify the pool of RNA sequences. Primers designed with unique nucleotide tags can also be used in this step to create ID tags in pooled library multiplex sequencing. Let's now venture into the realm of sequencing and explore the fascinating intricacies it holds. The actual RNA sequencing varies significantly depending on the platform used. 3 common next-generation sequencing platforms are pyrosequencing on the 454 Life Sciences platform, polymerase-based sequence by synthesis on the Illumina Company platform, or sequencing by ligation on the ABI solid sequencing platform. As we progress through this video, let's now turn our gaze towards data analysis. Central to miRNA-seq data analysis is the ability to 1. Obtain miRNA abundance levels from sequence reads, 2. Discover novel miRNAs and then be able to 3. Determine the differentially expressed miRNA and therefore associated miRNA gene targets. Now, let's dig deeper into miRNA alignment and abundance quantification and unveil the hidden treasures it holds within. miRNAs may be preferentially expressed in certain cell types, tissues, stages of development, or in particular disease states such as cancer. Since deep sequencing miRNA-seq generates millions of reads from a given sample, it allows us to profile miRNAs, whether it may be by quantifying their absolute abundance, to discover their variants known as isomers. Note that given that the average length of sequence reads are longer than the average miRNA 1725 and the 3 and 5 ends of the miRNA should be found on the same read. There are several miRNA abundance quantification algorithms. Their general steps are as follows. 1. After sequencing, the raw sequence reads are filtered based on quality. The adapter sequences are also trimmed off the raw sequence reads. 2. The resulting reads are then formatted into a FASA file where the copy number and sequence is recorded for each unique tag. 3. Sequences that may represent yeast coli contamination are identified by a BLIST search against an yeast coli database and are removed from analysis. 4. Each of the remaining sequences are aligned against a miRNA sequence database such as miRNAs in order to account for imperfect dicer processing, and overhang on the 3' end and on the 5' end are allowed. 5. The reads that do not align to the miRNA database are then loosely aligned to miRNA precursors to detect miRNAs that might carry mutations or those that have gone through RNA editing. 6. The read counts for each miRNA are then normalized to the total number of mapped miRNAs to report the abundance of each miRNA. As we progress through this video, let's now turn our gaze towards novel miRNA discovery. Another advantage of miRNA-seq is that it allows the discovery of novel miRNAs that may have eluded traditional screening and profiling methods. There are several novel miRNA discovery algorithms. Their general steps are as follows. 1. Obtain reads that did not align to known miRNA sequences and map them to the genome. 2. RNA folding method 1. For the miRNA sequences where an exact match is found, obtain the genomic sequence including a flanking sequence on either side and run the RNA through RNA folding software such as the Vienna package. 2. Folded sequences that lie on one arm of the miRNA hairpin and have a minimum free energy of less than xmol are shortlisted as putative miRNA. 3. The shortlisted sequences are trimmed down to include only the possible precursor sequence and are then refolded to ensure that the precursor was not artificially stabilized by neighboring sequences. 4. The resulting folded sequences are considered novel miRNAs if the miRNA sequence falls within one arm of the hairpin and are highly conserved between species. 3. Star-strand expression method MIRDEEP 1. Novel miRNA sequences are identified based on the characteristic expression pattern that they display due to dicer processing, high expression of the mature miRNA over the star-strand and looped sequences. The spotlight now falls on differential expression analysis as we delve deeper into its details. After the abundances of miRNAs are quantified for each sample, their expression levels can be compared between samples. One would then be able to identify miRNA that are preferentially expressed at particular time points, or in particular tissues or disease states. After normalizing for the number of mapped reads between samples, one can use a host of statistical tests like those used in gene expression profiling to determine differential expression. Get ready for an exciting part as we dive into target prediction. Identifying a miRNA's miRNA targets will provide an understanding of the genes or networks of genes whose expression they regulate. Public databases provide predictions of miRNA targets, but to better distinguish true positive predictions from false positive predictions, miRNA-seq data can be integrated to miRNA-seq data to observe for miRNA functional pairs. miRNA targets can, Miranda, and Picta are software designed for this purpose. A list of prediction software is given here. The general steps are: 1. Determine miRNA binding pairs, complementarity between the miRNA sequences at the 3 UTR of the miRNA sequence is identified. 2. Determine the degree of conservation of miRNA binding pairs across species. Typically, more highly binding pairs are less likely to be false positives of prediction. 3. Observe for evidence of miRNA targeting in miRNA-seq or protein expression data. Where the miRNA expression is high, the gene and protein expression of its target gene should be low. In this section, we'll be exploring target validation for cleaved miRNA targets. Many miRNAs function to direct cleavage of their miRNA targets. This is particularly true in plants, and thus high-throughput sequencing methods have been developed to take advantage of this property of miRNAs by sequencing the uncapped 3 ends of cleaved or degraded RNAs. These methods are known as degradome sequencing or PARE. Validation of target cleavage in specific RNAs is typically performed using a modified version of 5 rapid amplification of ends with a gene-specific primer. As we enter this new phase, let's analyze identification of novel miRNAs from different angles and evaluate its significance. miRNA-seq has revealed novel miRNAs that were previously eluded in traditional miRNA profiling methods. Examples of such findings are in embryonic stem cells, chicken embryos, acute lymphoblastic leukemia, diffuse large B-cell lymphoma and B-cells, acute myeloid leukemia, and lung cancer. Let's now turn our attention to disease biomarkers and uncover the fascinating insights it brings to the table. MicroRNAs are important regulators of almost all cellular processes such as survival, proliferation, and differentiation. Consequently, it is not unexpected that miRNAs are involved in various aspects of cancer through the regulation of onco- and tumor suppressor gene expression. In combination with the development of high-throughput profiling methods, miRNAs have been identified as biomarkers for cancer classification, response to therapy, and prognosis. Additionally, because miRNAs regulate gene expression, they can also reveal perturbations in important regulatory networks that may be driving a particular disorder. Several applications of miRNAs as biomarkers and predictors of disease are given below. Cancer type miRNAs ref. Width equal 90 spin equal 3 styler text align. Left breast ER status miR-EOB, miR30 family, miR-ERB, miR155, miR342, miR206, miR191 PR status letic, miR-ERB, miR-ER, miR30 family, miR-ERB status miR-ERD, miR-ERK, miR-ERK, miR-ERB, miR-I width equal 90 spin equal 3 styler text align. Left lung squamous versus non-squamous cell miR205 small cell versus non-small cell miR17 IP, miR22, miR24, miR31 width equal 90 spin equal 3 styler text align. Left gastric diffuse versus intestinal miR-ABIC, miR30 family, miR-EOB width equal 90 spin equal 3 styler text align. Left endometrioid versus uterine papillary miR-EOB, miR-IIP, miR101, miR452, miR382, miR-ER, miR width equal 90 spin equal 3 styler text align. Left renal clear cell versus papillary miR424, miR203, miR31, miR126 oncocytoma versus chromophore miR-ARC, miR139 IP width equal 90 spin equal 3 styler text align. Left myeloma with miR1, miR-ER with miR203, miR155, miR375 with miR-ER, miR650, miR184 with equal 90 spin equal 3 styler text align. Left acute myeloid leukemia with miR382, miR134, miR-ER, miR127, miR299 IP, miR323 with orange letterbic, miR127 with mutations miR-EOB, LET7, miR29, miR204, miR-ER, miR-EOB with foot ITD miR155 with equal 90 spin equal 3 styler text align. Left chronic lymphocytic leukemia's EAP70 levels and its status miR-ER, miR195, miR221, miR155, miR-UP with equal 90 spin equal 3 styler text align. Left melanoma with BRAF miR-ER, miR338, miR565 with equal 90 spin equal 3 styler text align. Left lymphoma diffuse large B-cell lymphoma has miR128, has miR129 IP, has miR152, has miR155, has miR185, has miR-ER IP, has miR-UB, has miR-UB IP, has miR-UB, has miR-ER, has miR-ER, has miR28 IP, has miR-ER, has miR331 IP, has miR365, has miR625, has miR9. Styler text align. Left table 1, cancer subtypes distinguished by MicroRNAs. Earth's is not a comprehensive list of miRNAs involved with these malignancies. Let's now turn our attention to comparison with other methods of miRNA profiling and uncover the fascinating insights it brings to the table. The disadvantages of using miRNA-seq over other methods of miRNA profiling are that it is more expensive, generally requires a larger amount of total RNA, involves extensive amplification, and is more time-consuming than microRNA and CRISPR methods. As well, miRNA-seq library preparation methods seem to have systematic preferential representation of the miRNA complement, and this prevents accurate determination of miRNA abundance. At the same time, the approach is hybridization independent and therefore does not require a priori sequence information. Because of this, one can obtain sequences of novel miRNAs and miRNA isomers, distinguish sequentially similar miRNAs, and identify point mutations. Get ready for a captivating exploration as we unravel the layers of platform comparison of miRNA profiling and their profound significance. MicroRNA sequencing throughput time hours days weeks total RNA required 500 eng 100 1000 eng 500 5000 eng dynamic range detected 6 orders of magnitude 4 orders of magnitude 5 or more orders of magnitude infrastructure and technical requirements few moderate substantial cost per sample USD $400. Styler text align left table 2 platform comparison of miRNA profiling. Thanks for being a part of this amazing journey. I can't wait to bring you more exciting content.